Dear students, Assalamu alaikum. Today I will deliver a lesson for class 12th subject zoology. Since in our previous topic we discussed about the linkage, its discovery and the chromosomal theory of linkage. And today we will discuss about the types of linkage. So far the types of linkage are concerned, there are two types of linkages. Number one is the incomplete linkage and number second is the complete linkage. Now first of all we will discuss about the incomplete linkage. Now when genes are present on different chromosomes or on the same chromosome distantly, that is apart from each other, they have a tendency to occasionally separate due to crossing over. They produce a dihybrid ratio of 9:3:3:1 and a test cross ratio of 1:1:1. That is, they produce a few recombinant types in addition to the parental types. Now this incomplete linkage was demonstrated in female Drosophila. When a red-eyed and normal-winged homozygous female Drosophila was crossed with a homozygous recessive purple-eyed and vestigial-winged male, the progeny, that is the F1 generation, are all heterozygous with red-eyed and normal-winged. Now when F1 females flies or Drosophila are test crossed with the homozygous recessive male, it does not yield the ratio of 1:1:1. Instead, the ratio comes out to be 7:1:1:7. This shows that the two genes did not segregate independently of each other. And this type of linkage in which non-parental combinations are also formed is called incomplete linkage. It is basically due to the crossing over. The number of recombinant individuals is usually less than the number of expected in independent assortment, and the frequency of parental form is much higher, that is 87.4%, than the recombinant, that accounts for 12.6% only. Now this shows that two genes did not segregate independently of each other and only 12.6% recombinant types were observed, which is quite different from 50% recombinants of independent assortment. Now it also indicates that in the oocyte of F1 generation, only some of the chromatids cross over while the majority is preserved intact. That accounts for 87.4% parental types in the progeny. Now this was the incomplete linkage. Now we will discuss about the complete linkage. Now when the genes are located on the same chromosome, they do not separate during inheritance. They are always inherited together due to absence of crossing over. This complete linkage is rare but has been reported in Drosophila. For example, when a red-eyed normal-winged female is crossed with a purple-eyed vestigial-winged male Drosophila, the F1 generation produces the heterozygous ABAB. Now when this F1 generation heterozygous are test crossed with a recessive parent such as AB and AB, the gametes produced are such as AB, AB and AB. The ratio of these gametes remains 1:1. So, a red-eyed and normal-winged homozygous female Drosophila, that is AB-AB, is crossed with the homozygous recessive purple-eyed and vestigial-winged female Drosophila. The progeny or F1 generation individuals are heterozygous with red-eyed and normal-winged phenotypically. Now, when F1 males are test-crossed to a homozygous recessive female, purple-eyed and vestigial-winged, only two types of individuals are produced, that is, with red-eyed normal-winged and purple-eyed vestigial-winged in the ratio of 1:1. That is, the parental phenotypes only. Similarly, during inbreeding of F1 generation, recombinant types are always absent. This indicates that there was no crossing over, which accounts for the complete linkage in male Drosophila. Now, this was all about the types of linkage. In our next topic, we will discuss about the linkage groups as well as the importance and significance of linkage. Until then, goodbye.